Welcome to our presentation on type 1 diabetes mellitus, a novel approach to insulin expression, by myself, Mona, and my colleagues Amira and Batria. Type 1 diabetes is characterized by the autoimmune destruction of pancreatic beta cells and often occurs in children and adolescents. Normally after eating, sugar in the form of glucose from the food enters the bloodstream. The hormone insulin is then released from the beta cells in the pancreas in response to the rising levels of glucose in the blood. However, as beta cells are destroyed in type 1 diabetes, little or no insulin is produced. Insulin is responsible for transporting the glucose into the body's cells to be used as vital sources of energy. It does this by binding to insulin receptors, which triggers the opening of glucose channels, allowing glucose to enter the cells. Once inside the cell, blood glucose levels decrease. In a healthy pancreas, low blood glucose levels signals the beta cells to lower the amount of insulin secreted, thus decreasing the amount of glucose entering cells and preventing low blood sugar levels, also known as hypoglycemia. The destruction of the beta cells that occurs with type 1 diabetes throws this entire process into disarray, as insulin is not readily available to signal the uptake of glucose. As the glucose cannot enter the cells, for example those found in muscle and adipose tissue, they starve for nutrients, and other systems in the body must provide the energy needed for normal functioning. Individuals with this condition rely on the supplementation of an external source of insulin with the use of daily insulin shots or an insulin pump. These treatments tend to inaccurately match the body's physiological need and therefore cause the glucose to fall below normal levels, leading to the unpleasant symptoms of hypoglycemia. Finding out a possible approach for the internal production of insulin would provide a plausible solution to this problem. As insulin expression is largely restricted to pancreatic beta cells, much of the research into type 1 diabetes mellitus tends to target these cells. It has been found that there are low levels of insulin expression in extrapancreatic tissue, such as the brain and the thymus. Knowing that non-beta cell types can produce insulin, regardless of how insignificant the level, is very important and gives rise to many questions. Can we somehow upregulate or induce insulin expression in non-beta cell types as a possible alternative to current type 1 diabetes treatment? And if so, what type of non-beta cell types would be the most efficient? If insulin expression is eventually induced in non-beta cell types, it is important to use a cell that effectively releases the insulin into the bloodstream so that it can be utilized. Alpha cells are attractive candidates because of their secretory nature, their developmental proximity to beta cells, and their location within the islets of Langerhans. In the next few slides, my colleague Amira will discuss a recent publication which aims to use alpha cells in this way.
GW8510 increases insulin expression in pancreatic alpha cells through activation of p53 transcriptional activity. This investigation explores the previously mentioned concept of insulin expression in non-beta cell types using mouse alpha cells. Mice have a two-gene system, INS2 and INS1, unlike the human insulin system, which only has one. However, INS2 is very similar to the human insulin gene, which is why the team was focusing on the modulation of INS2 transcription in mouse alpha cells. From previous research, which indicates the role of GW8510, a small molecule that acts as a putative cyclin-dependent kinase 2 inhibitor, the team hypothesized that small molecule-mediated stimulation of insulin expression in alpha cells is a necessary initial step for insulin production. Now the team wanted to examine the specific mechanism involving the role of GW8510. Mouse pancreatic alpha cells, also known as alpha TC1, and human islet cells were treated with GW8510 for 5 days. A dose-dependent expression of INS2 was observed. The levels of glucagon, which is the hormone normally expressed by alpha cells, remained unaffected. This is important. The main aim of the experiment, which is to induce insulin expression in alpha cells, should not affect the original role of these cells. GW8510 was able to induce the expression of INS2 in 48 hours, as seen in Figure 1. The induction, however, did not occur directly, but through another mechanism. they were able to demonstrate involvement of the p53 signal transduction pathway in the modulation of INS2 expression levels. p53 has transcription factor activity and performs most of its biological functions through direct regulation of downstream transcriptional targets such as CDKN1A, MDM2, and CCNG1. Through real-time PCR, they were able to confirm that p53 transcriptional target levels were upregulated by treatments with the compound. The team thus sought to examine said mechanism by which GW8510 activates the p53 pathway and upregulates INS2 expression. By examining pathways upstream of p53, they discovered that both SP600125, a JNK123 inhibitor, and SB202190, a p38 kinase inhibitor, could almost completely suppress the induction of INS2 by GW8510. It was clear that the JNK and p38 pathways were also crucial for GW8510 to induce the expression of INS2. If that was not the case, the introduction of SP600125 and SB202190 would have no significant effect on INS2 expression. In the next few slides, my colleague Badria will discuss how understanding these pathways could be applied to possible treatments for type 1 diabetes.
The team observed a high p53 protein level in the alpha cell line, indicating that modulating p53 activity in alpha cells is a feasible strategy to induce insulin expression. However, the p53-dependent insulin induction by GW8510 in pancreatic alpha cells does not occur in non-pancreatic cell types because they are not specialized to secrete insulin. GW8510 does not appear to act at the level of p53 directly, but acts upstream of p53 to target JNK and p38 dependent signal transduction pathways. Within a cell's nucleus, regulatory elements in DNA called promoters and enhancers communicate with each other to carry out gene expression over large genomic distances. And because the promoters and enhancers physically touch each other, the DNA sequences bend into loops made of chromatin fiber. It remains uncertain as to whether these DNA loops are involved in gene transcription, and a detailed exploration of looped genomic interactions at the insulin promoter may yield further insights into the regulation of insulin expression in pancreatic beta cells and the potential for upregulating insulin expression in a non-beta cell type. Evaluation of this prediction will help determine whether modulation of p53 activity could also be used to alter insulin expression in human endocrine cell types. Diabetes affects many millions of people worldwide. The optimal treatment is to restore the endogenous supply of insulin, either through transplantation of the pancreas or transplantation of islets of Langerhans or beta cells. However, the donated pancreas organs are limited, and the available organs are only able to treat a small portion of the diabetes patients. Thus, glucose-responsive, insulin-producing cells from human origin are urgently needed. This type of research brings us one step closer to a treatment that would match the actual physiological needs of the body, as hypothetically a patient's non-beta cells could produce the insulin the body needs. We hope this presentation has shed some light on possible innovative approaches to treating type 1 diabetes mellitus.